we evaluated HDAC8, which is causes the deacetylation of lysine residue on the N-terminal part of the core histone and non-histone proteins. There are many HDACs. We focused on one HDAC. Now, HDAC8 we found is overexpressed in myeloma, and it is connected, its expression is connected with survival outcome. And that led us to do the more detailed analysis. And what we identified is that when we inhibit HDAC8, either by genetic means or we have a drug that inhibits HDAC8, the myeloma cell viability is significantly impacted. Now, this inhibition of myeloma cell growth is also associated with some impact on genomic stability. We identified that if we inhibit HDAC8, it decreases DNA breaks in a time-dependent manner. So the spontaneous DNA changes that happen by inhibiting HDAC8, those don't happen. And if we inhibit HDAC8, it causes more breaks, but also at the same time, it increases homologous recombination, which is one of the key pathways that maintains the genome stability. So they both appear to be connected. Now, the way it impacts the homologous recombination, what we report, is that it impacts by deacetylating RAD51, which is the main player of the homologous recombination pathway. And so that looks like as the mechanism of action. And effect of this drug, the inhibitor of HDAC8, is also confirmed in an animal model that by its action directly on the cell growth and on the genome changes, it can also inhibit growth of myeloma cells in an animal model. And then it synergizes with some of the conventional anti-myeloma treatment. So it's a good inhibitor by itself. It has a synergistic activity. So what we report here is that HDAC is a good target in myeloma. It is responsible for causing some of the genomic changes that happens with evolution of the disease. And that if we inhibit with this agent, which is actually, it's a potential for a clinical grade material, we can kill myeloma cells on one hand, but also at the same time, in the surviving cells, we can decrease acquisition of new genomic changes. And I think that is very attractive. Synergistic effect with the standard combination provides us a rationale to investigate this in clinical setting.